In this video, we are introduced to our fourth and final macromolecule, and that is the nucleic acids. Nucleic acids store information for the ever-important life-supporting work of producing proteins. By the end of this video, you should be able to identify the two major nucleic acids, those are DNA and RNA, and be able to describe the 5 major nucleotides that make up those nucleic acids. We've examined 3 of life's macromolecules so far in this course. We've talked about carbohydrates, lipids, and proteins. So now we're going to turn our attention to the 4th macromolecule, perhaps one of the most important, and it is the nucleic acids. Nucleic acids are macromolecules that store information. And there are 2 types of nucleic acids. The first is the deoxyribonucleic acid, or DNA, and the second is just simply ribonucleic acid, which is RNA. The difference being deoxyribonucleic acid starts with the prefix deoxy, which tells us that there's something going on in that molecule that's a little bit different than the other type of nucleic acid, the ribonucleic acid. But both play a very central role in directing the production of proteins in living organisms. And by doing that, they play a central role in determining all of the inherited characteristics of the individual. Nucleic acids are made up of individual units called nucleotides. So the nucleotides are the monomer that makes up the polymer of nucleic acids. Now, all nucleotides have 3 components. They have a molecule of sugar, a phosphate group, and so this is a molecule that contains a phosphorus atom bound to 4 oxygen atoms, and then the third part of the nucleotide is the nitrogen-containing molecule. You can see that depicted in the picture displayed. In both types of nucleic acids, nucleotides are linked in a series to form this ribbon-like strand, and this is the backbone of the nucleic acid molecule. So you have the sugar molecule portion of the nucleotide is attached to a phosphate group, which is attached to another sugar, which is attached to another phosphate, and so on and so on. So attached to each sugar and protruding from the backbone is one of the nitrogen-containing molecules called DNA or RNA bases. And they're named this because of their chemical structure. A 10-unit nucleic acid strand, therefore, would have 10 bases, one attached to each sugar within the sugar-phosphate backbone. But the base that's attached to each sugar isn't always the same. It can be one of several different bases, and this is the reason that nucleic acid is often described by its sequence of bases. Nucleic acids store information in the order of the bases that are attached at each position in the molecule's backbone. At each position in a molecule of DNA, for example, the base can be any one of 4 possible bases. It can be adenine, thymine, guanine, or cytosine. We often refer to these based on the letter that each word begins with. So adenine would be A, thymine would be T, guanine would be G, or cytosine, and that would be C. Now, just as the meaning of a sentence is determined by which letters are strung together, the information in a segment of DNA is determined by its sequence of bases. One DNA segment may have the sequence adenine, 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 guanine, cytosine, thymine, guanine, which would be abbreviated by their letters A, G, C, T, G. Another DNA segment may have the sequence CGATTA, CCCGAT. Isn't that fun? Because the information differs in each case, so too does the polypeptide for which the sequence codes, as we're about to find out.